We can start with the last lecture with Professor Sanjay Jain. I think there won't be any further extra classes for sure. So I think he's still here today. I think if you have some time, if you have any questions, please get hold of him at the end of the class today and ask him. He will be away this weekend, so we won't be here next week. So we have had the privilege to have these 5 lectures, and we really enjoyed. So looking forward to the last lecture, sir.
In the last 4 lectures, what we did was, first 2 lectures, we did the puzzles and the empirical puzzles. In the third lecture, we discussed the bacterial growth laws, how they can be understood in terms of model. In the third lecture, we understood how these 2 items, self-reproduction, balanced growth, on the one hand, and exponential growth, can be understood in terms of models. In the last lecture, we did bacterial growth laws, and today we will do questions related to cell individuality. So far, we've been studying the average behavior of cells. Okay. Now we will study how fluctuations dominate. What happens when you have fluctuations.
You're saying average.
So those, because they are deterministic differential equations, and we are treating populations as continuous variables satisfying those deterministic dynamical equations. That is really capturing the average behavior of cells. Okay. So the variability between cells has something to do with stochasticity. And in the entire framework that I've talked about today, up to now, we've not really talked about stochasticity. So today we will introduce that and see how what that does to the dynamics and how that compares with experiment. So without much ado, let me turn to the model that I will be mainly discussing. This is just one model, okay, and is one model in a class, in a larger class of models, and there are related classes of models. Many of them share similar properties. So, but for its simplicity, I will present this model, and it actually explains a number of things that are observed. So. This is the familiar model, the PTR model, in which there are 3 variables, P, T, and R, representing these 3 pools. Okay, the precursors, the metabolic enzymes, and the ribosomes. Okay, there is this additional terms that have been added here: the death rates of the macromolecules, the degradation rates. Okay, it doesn't change anything material in what I've told you before about this model. So this is what I will refer to as the PTR sector of the model. Now, in addition, we will talk about another molecule, which is the so-called timekeeper molecule. This is the molecule whose population will govern cell division. Okay, so Z is the population of some other protein in the cell, and this protein also grows in a manner that is similar to our other proteins, namely the enzymatic proteins, the transporters and enzymes T, and the ribosomal protein R, ribosomal. Okay, so its population also. It's produced in as ribosomes basically convert the information on its messenger RNA, read the messenger RNA, and convert amino acids, which is P, into this protein. Okay, so this is catalyzed by R, uses a protein, and there is another rate constant here, which ultimately reflects how much messenger RNA there is for this particular protein. And this timekeeper protein, we will assume, Its numbers are small, much smaller than the numbers of things here. Okay, and so the volume, the volume of the system, which we had said was V is equal to little v into P plus T plus R. We could add another term Z here. Okay, but since these numbers are so small compared to this, it doesn't really matter. Okay, so for all practical purposes, we will continue to take the volume, whatever it is. Okay, before. That has a very great convenience that the X sector dynamics is completely independent of the Z sector dynamics. The Z sector dynamics, of course, depends upon this upon the X sector. This PT and R are very much here. Okay, so that's the general formulation. Okay, so I will. Exactly. Exactly. So it's a practical convenience here. And so, in fact, everything that I've said about this PTR sector before still holds. That it will get into an exponential growth attractor where the ratios of populations will become constant. Okay, volume will also grow exponentially at the same rate as all the populations. Okay, and so growth law can come out of it if you optimize the fractions here, there, and so on. So all the things that you could do with this sector, you can still do. Then changes. And now the only thing is that there is a timekeeper molecule, and the significance of this timekeeper molecule is the fact that we will declare that when its population reaches a certain threshold value, which we will call ZC, and that is a parameter of the model, another new parameter of the model. When it reaches that value ZC, the cell will divide. The cell takes a decision to divide. Okay. Now, okay, let me. Yeah. So, this particular model is again a part of a larger class of models in which you have this X sector as before with N chemicals, and then there is the Z sector which just has a single chemical Z. Yes? Question? Yes.
So in the PTR dynamics, KT plus KR will be equal to small k.
So KT and KR will, so KT multiplied by MT plus KR multiplied by MR will be equal to small.
Okay.
Yeah. Right. Okay. So, so you can have a structure of this kind, and V, Fi, and H are taken to be homogeneous degree one functions of the X variables. Okay. So this is the dynamics that describes cell growth, and cell division is governed by the Z sector. So the Z sector affects everything else only through division. Okay. And what's it? That we say that these equations hold as long as Z is less than ZC, which is the threshold. Okay. And that's the division threshold. And when Z becomes equal to ZC, then cell divides, and all the X's halve. They become half their original values, like we did yesterday. Some examples, and Z can also halve, but we will allow something slightly more general than that. That Z goes to some reset value, okay, which is the value of Z at birth. Okay, in this case, okay, so it's a reset here. And then when the cell starts, it starts with Z is equal to ZB, and Xi, half the Xi is at the time of division, and then this dynamics takes over again. Okay, the growth dynamics takes over again, You follow this dynamics until Z has again become ZC, and then you divide the cell and so on. Okay, so that's what you are.
Yeah, you said the small k is only related to Kp and Kt and Kr.
Yes.
But why it is not taking account that Z factor also? Because P is decreasing when Z is
Yes.
yeah. So
Okay, good point. Yes.
so in the K info, so small K information.
Absolutely.
That should,
Okay, good question. Indeed, there should, because Z is increasing,
right. So P,
so there should be a term here. I mean, this K should account for that.
right.
So in answer to my question there, I should have included a factor of,
Right.
of this production.
Right.
So, in principle, that's absolutely correct. But the logic is the same as the one that I used here. The amount of Z molecule is so small
Okay.
that it doesn't really matter to this whole dynamics. So it's essentially, I mean, as far as the P production is concerned, yes, some P will be depleted in this, and some R will be allocated to make Z, but those are so small that you can neglect. Okay, so the Z sector is a very is a sector which we are as an approximation here. Okay, you can of course do more complicated things and have those things here, but as an approximation, we are treating this the Z sector independently.
So ZC and ZB are also very small numbers.
Yes,
The critical.
indeed. Z is much smaller than for example, the values that P, T, and R will go to. Okay.
Okay.
So I'll give you an example. Okay, of this. What is this Z molecule? So we don't know, but there are a couple of candidates in E. coli for the Z molecule. There is a protein called DNA. There's a protein called DNA A. Okay, this is a protein. This is the protein that is known to trigger the replication of the DNA molecule. Okay? So what is the DNA molecule? The DNA molecule actually is a circular molecule, double-stranded circular molecule. Okay, inside the cell. This is in those pictures that I showed you. This is that yellow rope, right, which is going all over the cell and so on. So, right, that is the DNA molecule represented like this. And somewhere on the DNA molecule is a location which is called ORI-C. Okay. Which is the place where the origin of replication happens. Okay. This is the place where the DNA polymerase, which is the enzyme that makes 2 copies of the DNA, replicates the DNA. That's where, this is the site at which that enzyme, the DNA polymerase, so DNA polymerase, so DNAP, let me call that. That's a protein molecule which binds here and starts replicating the DNA. And this whole DNA polymerase complex, this starts moving in 2 directions, here and here, and you start making a copy of the DNA molecule. Okay. And this DNA polymerase moves here with a certain speed in a certain time. It finishes the replication. And then the 2 copies of the DNA molecule, they segregate, okay, spatially segregate. And then the cell gets pinched in the middle, in which such that the 2 halves, the 2 daughter cells, each has 1 copy of the DNA molecule. And of course, all the other molecules that are there inside the cell. Now, this DNAP molecule can't just latch on to this ORI-C molecule. Okay. The trigger for it to latch onto this molecule is provided by this DNA protein. Okay. So this is the triggering molecule, and what it does is that it is known So this DNA A protein, it gets phosphorylated. So the protein is produced. It gets attaches a phosphate group is attached to it, and that's called the so-called active version of the DNA protein. And this active version then binds. So this is actually a location. So if I open it out, okay. So this is actually a location somewhere. Okay, this is a region in there. And what happens is that this DNA A protein, which is the active version of it, it actually latches on to this place. And it is known that DNA polymerase is recruited to cause the self-replication, to cause the replication of the DNA molecule, when the number of these active DNA A molecules, so this is DNA A active, okay? When these molecules, about 20 to 30 of these molecules, when they bind here, okay? So when they bind here, basically, okay, that is when the DNA A is recruited. That is when the DNA polymerase is recruited and replication starts. And the moment the replication starts and the DNA polymerase starts moving in the two directions to replicate the DNA molecule, this complex of 20 to 30 DNA A active molecules sitting at the origin site, okay, it dissociates. Okay, it falls apart. So you can consider, the idea here is that you think of the Z population as this population. Okay, so this is what we are talking of. You can think of this as the Z, which is the population of the active DNA A molecule sitting at the site for this. And these these molecules, they dissociate, okay, and then they start building up again. This whole process of DNA A active attaching itself here starts again. When it reaches the that critical value, okay, 20 to 30, okay, then the next round of replication starts. Okay? That's what is happening here. So this is one of the molecules that is a candidate for this timekeeper molecule. Yeah. Yes.
So this replication form is starting in both sides, right? And this start of replication is not coordinated with the cytokinesis, right? That is the division of this legular membrane. So this is continuously happening. So your simple model is sort of reconciling with that, or
yes. So the simple model is assuming that cytokinesis, which is the process of the actual pinching of the cell and dividing, okay, that is a slave to this process. Okay, so that is you can think of so this is what is triggering replication, and then in due course of time, cytokinesis will happen. So after this, the cell will pass through what are called the C and D phases of its growth. The C phase being the replication phase, and the D phase being the part where it segregates and actually stops. And that is happening, as a slave after a certain time interval, let's say, okay, a fixed time interval after this replication starts. Yes. Yeah, thank you.
These active molecules, they're getting attached to.
That's this.
Oh,
this is the region of the DNA.
Okay, so first they are attaching, and once that number of these molecules reaches this threshold value,
So then it triggers the other guy, the DNA,
The DNA polymerase. It
is to come
triggers it to be recruited.
and then to start the replication process.
Yes, and it is associated with certain actions in which the shape of the DNA molecule there is modified and so on, and this one attaches, and that one then dissociates. Once that attaches and starts its job, this one dissociates.
And this number is what you would be calling Z.
So this, the number that is needed here is what I will call ZC. Okay.
The 20/30.
That's right. Yeah, that will be my ZC. Okay.
Okay.
So you can see that this number is much smaller than the other molecules in the game. Of course, this is the number of active attached molecules here. The actual number of Z molecules is larger because they're there in the cytoplasm, and the inactive and active forms are all there. But nevertheless, it's a very small contribution to the overall cellular populations. Okay, and also that this constant ZB in this example should be 0, because this whole structure dissociates.
Okay.
So the moment it does its job, it dissociates. Okay, and then the replication starts. And the reason why it dissociates is that if it did not, immediately after this round of replication, a second round of replication would start. You don't want replication to be continuously happening. You need to make the right amount of DNA for the cell. Yeah. Okay. So it happens after a while, and that is governed again by the time that it takes for it to build up. Yes. In the case of. Yes. So that happens in rich media. When the bacterium is in normal conditions or starved conditions, then it typically has one DNA molecule, one copy of the DNA molecule at birth. At birth, it has one copy, and there are no forks. And at some point, when the DNA builds up onto this ODC, then it starts replicating. And then it makes two copies, they segregate, and then that happens. But when, just to address your question, when there is, this process of replication of the DNA itself takes 50 minutes or something like that. Okay. But we know that in good media, DNA can actually divide in less than 50 minutes. It can divide in 20 minutes, right? So how can it do that? The question is, how can it do something like that if it takes 50 minutes for this to replicate itself, right? Then how can the DNA divide in 20 minutes, right? And that is where the multiple fork hypothesis comes in. That, before this round of replication is complete, this has built up again at ODC, okay, right? And a second round of replication starts, so that you have multiple forks within forks. I mean, of replication. However, when a replication round gets completed, when a round that has started at any particular point gets completed, the two parts of the DNA do segregate. So, along with all their existing forks, there is a segregation. Okay, so the DNA that is born in that case is born with all, the two DNA, each of them have those forks. And, this replication is continued. Okay, it's not that the cell starts it. So that's the process. Okay? Yeah.
It's just really helpful for this non-biology audience to know that the DNA is only circular in bacteria and in mitochondria and chloroplast, and it's linear in eukaryotic nucleus. So I hope everybody is aware of this.
That's right. This mechanism is only known in bacteria, and we don't know. It's still not settled that this is the protein that triggers division. Okay, what triggers division? We still don't know. Okay, but this seems to be one candidate. Another candidate is the FTSZ molecule itself, which pinches, which forms a ring near the center of the E. coli rod-shaped cell, near the center of the cylinder, and then pinches. And that also, it might be that there is a threshold that you need for that molecule, and that molecule, then pinches. So that is another candidate, okay, for this process. But I'm giving you the motivation. So for this to be equal to 0, okay, for the reset value to be 0. So I've kept this here in the slides as a parameter because you can do an analysis in general, but for many purposes we'll just take this ZB to be 0. So now one has defined this growth division dynamics. The growth part and the division part both are defined. Parameters have to be specified, ZC, ZV, KZ, etc. The model is then completely specified, and you can put it on the computer. And this is what you see. Okay. So now P, T, and R are in green, blue, and orange. These are done with these parameter values, which are kept with an eye on actual parameters that might obtain inside the cell. So these populations are into 10 to the 7. So P is 10 to the 7. Okay. So these numbers, because the number of P molecules is much, much larger than the number of T molecules and the number of ribosomes. In order to show them on the same figure, they have been rescaled all differently. Okay. All of them have been rescaled differently. Okay. So so Z has been rescaled the most, okay, and basically in the red molecule is the Z1, and it is going from 0 to about 25, okay, and then it is dropping to 0, okay, then B is 0, that's the kind. Okay, and you can start from any initial condition. You will end up in the same steady state. That's what these two pictures show. Two different initial conditions. And you see that the PT and R molecules, when you plot them on a semi-log plot versus time, the growth parts are linear, which means that they have exponential growth, and that exponential growth slope is the same as what was there earlier in the PTR model. Okay, that doesn't change. Okay, the ratios become constant. But notice that the Z dynamics is not exactly exponential. Okay. Even though this is a homogeneous linear function, homogeneous degree 1 function of the X molecules, it's not, and we'll see why. The reason is that you are not halving in this model. One is not halving the Z population. One is taking it from some value and pushing it away, to 0. Okay, so that's why it's not exponential. But anyway, so that's what it is, and the volume grows exponentially. The volume also doesn't matter what the initial condition is, settles down into the attractor in which it grows exponentially. And halves and so on, and the concentrations reach a fixed point. So that's what you expect. Now, what one is interested in is what happens when you allow stochasticity in the dynamics. Okay? So this is the behavior, and every cell will behave like this. According to this model, every cell will be like this. Every cell at birth will have this size, right? At division will have this size. Will take exactly the same time to divide. Okay? So there is no variation from cell to cell in this model. It's a deterministic model. But we know that chemical reactions actually happen stochastically. Right? There is a list of reactions that any reaction can happen depending upon which chemicals are there, and which reactions happen first, which reaction happens second, and so on, that is completely probabilistic. Okay. Also the populations of molecules are not continuous variables. And here we've treated all the 4 populations as continuous variables so far, but actually they are integers, okay? 0, 1, 2, 3. So, and therefore those populations, depending upon which reaction happens when, they are changing in an integer fashion, but they are changing stochastically. The time dependence, the trajectory of the population of each molecule is no longer smooth, but it's going to be jagged, right? The molecule is going to be produced, then another molecule is going to be produced next time, and so on. So let's build that in. And you can build that in using what is called the Gillespie algorithm. So we are no longer simulating the deterministic differential equations. We are using a stochastic simulation to update the populations. And when you do that, those very same curves become stochastic. Okay? Each of these curves, actually P, T, R, and Z, they are actually stochastic. Okay, each of these growth parts are stochastic, but because the numbers of P, T, and R are relatively large, you don't see the stochasticity very clearly in these plots. But in the Z, you can see it more clearly. You can see the jaggedness with the numbers of Z from 0 to 25. Right? Is this point clear? So you can see that. And now you see multiple things. You see that Z takes sometimes a smaller amount of time to reach 25, sometimes it takes a larger amount of time to reach 25 because it's stochastic. Yes. Sorry? So yes, all the other populations are resetting to half, but Z is resetting to 0. Okay. So it goes from 0 to ZC, which is for this example was taken to 25. And so you see that the amount of time that Z takes to reach 25 is no longer fixed, but is now stochastic because the production of Z is a stochastic thing. Okay. And so this very naturally tells us that these interdivision intervals, the time intervals for which the cell grows after birth, have now become stochastic. And the stochasticity, so what is the stochasticity is set by the first passage time of Z. So Z is a molecule, and its first passage time to reach from 0 to ZC is now no longer deterministic as in the previous models, but now has become stochastic. And so the distribution of interdivision times is nothing but the first passage time distribution of the Z molecule. Okay? Is this point clear? First passage time means the time when the molecule Z, which is increasing, which is being produced according to its dynamics, according to its rates, okay, according to the reactions, the single reaction which it is being produced. This Z molecule will reach its critical value at different times in different generations, and therefore you will have a variation of the interdivision time. And because you will have a variation of the interdivision time, the growth, each of these populations, the big populations are growing exponentially with time, approximately, okay, even though they are also stochastic, but, because their numbers are large, the deterministic model applies reasonably well to them, and therefore, they are growing exponentially in every generation. The Z dynamics is not touching them in any generation. Okay, they are independent by assumption. Okay, of the Z dynamics. So once a division has happened and a cell has been born, they are growing exponentially. All that is happening is that how long they will grow exponentially is being cut off by Z, and that's why it's called the timekeeper molecule. So that's being cut off, and therefore, how much the cell will grow has also become stochastic, right? So the size. So if it's cell size starts at some size at birth, because the time for which this e to the mu t factor has to be applied. Okay, p of t, for example, is p naught e to the mu t. This is the deterministic version. In the stochastic, this is also not the correct equation, but the stochastic version of this. But approximately, if you imagine this one for p b e to the mu t, okay, and this time, and so the cell is going to go for a certain time t, and that time t is stochastic. That's the first passage time of z. So therefore, this factor is going to be stochastic, but the division volume is going to end up being stochastic. Okay, and if the division volume is going to be stochastic, the cell divides into half basically. Okay, the birth volume will also be stochastic. So all the macroscopic variables that describe the cell will become stochastic if they are slaves, if the cell division is a slave to this molecule Z. Okay? So the stochasticity in this, stochasticity in the size and all these big populations is arising because cell division has become slave to a molecule whose numbers are small, 0 to 25. Okay, because those numbers are small, therefore there is a stochasticity. You can roughly expect that if Z is going from 0 to 25, right, the amount, the coefficient of variation in the first passage time is going to be governed by the magnitude of ZC, which is 25. And it's going to be 1 over root ZC, square root of n, 1 over square root of n. Okay, so that is what will determine it. And so those microscopic fluctuations of Z have gotten imprinted on the macroscopic cell, okay, through this mechanism. So that's what's happening. And this, of course, Javier, I had shown you this experimental curve of what actually people see in experiments. This is the length, cell length versus time across different generations. This is a particular protein, fluorescence protein number with time. And these curves, so now the curves that we get in the model now start to resemble the experimental curves. Okay? Yes.
So the first passage time density that you have plotted for Z, right?
Yes.
The slide before,
Yes.
right? Shouldn't be the same for the others because I thought Z, when it reaches a critical value,
Yes.
the others also goes back to their half, like P goes to, right? Correct?
Yes. Yes.
So so is that stochastic time is controlled by only one distribution?
The time is governed by Z.
Yeah. What is plotted here? Like 3 different colors.
In this curve?
Yes.
Here?
Yeah.
Yeah. So what we are plotting here is the first passage time of Z.
Oh, that's the one for all of them. The inset one is basically the first passage time density.
Yeah. You're
Yeah.
pointing this out.
Yeah.
So the B subscript refers to birth.
Right.
Okay. And the D subscript refers to division. Okay. So VD is equal to volume of cell at division. And what this is? This will be simply V plus PD into PD plus TD plus RD. Okay. So that's if you
These values are stochastic.
yeah, so these values are stochastic, and then they're stochastic because okay, so this equation implies that PD is equal to PB e to the mu tau. Okay, and from generation to generation, this tau is different. Correct. Okay, therefore PD is different, and because in the next generation, PB is half of PD of this generation, therefore PB also becomes stochastic. Okay, and its distribution is given by this red one.
Yeah.
And you can also ask what is the added volume distribution. So you can also look for the distribution. So delta, the added volume is just VD minus VB. That's the added volume distribution. You can do that. And that is the distribution.
Yes. Yeah. And any other information on the nature of the distribution apart from for the first passage time?
Yes. So I will give you an exact formula for this
Nice.
and an exact formula for the delta distribution. Okay. And compare that with the experiment. Yeah. Okay. So yeah. So these are simulations. Yeah. So just to remind you of what I had told you earlier in the context of cell-to-cell variation. These for the, these people experiment. This is the generation time distribution for different media. The red is the rich medium. This one is the poorest medium. In rich medium, the cell divides fast. The mean generation time is smaller. In poor media, the cell takes longer to divide. The mean generation time is longer. But these are the distributions that are the observed distributions, for these cells in different media as they progressively go richer. And these are the division size distributions. In rich medium, the cell is larger. In poor medium, the cells are smaller. Okay, and those are those distributions. And what has been seen, division length distribution is such that the division length is correlated with the birth length. The interdivision time is negatively correlated with the birth length. The added size is uncorrelated. Okay? That's the so-called adder property. And is the value of ZC. How large do you allow it to grow? Okay. The larger ZC is, the narrower is the distribution. The smaller ZC is, the wider is this distribution, because fluctuations are governed by 1 over root n. Okay. And therefore, the larger n is the number of molecules. Okay. So the larger the n is, are the fluctuations. Okay. So you can see that distributions with ZC is equal to 20 are broader, and distributions with ZC is equal to 60 are narrow. That's a standard feature. Okay. And you can see that it's this range of ZC, 20 to 60, that fits the observed distributions. Okay. And this is interesting because this 20 to 30 is in fact the observed range of this complex size for triggering division. Okay. So it's nice that the numbers that are fitting the observed values of these size distributions are precisely the ones that for this DNA A molecule coincide with the range in which we know that the threshold, this association of ZC. So this ZC should be put to about 20 to 100, 10 to 100. Okay, let's say 10 to 100. Okay, something like that. That's in that range. Now, so that's that, and you can make an approximation in which you can exactly calculate the first passage time distribution. Okay. This approximation is one in which you say that the PTR dynamics, you take it to be deterministic. You say that those numbers are so large that the growth dynamics in any generation, okay, the birth volume of that generation, the birth populations of P, T, and R are going to be stochastic. But from birth to division, the growth dynamics is going to be governed by the ordinary differential equations that we had earlier. Why? Because the numbers of P, T, and R are quite large. Okay? P is of the order of 10 to 6, 10 to 7. T is several tens of, hundreds of thousands. And ribosomes are also in tens of thousands and so on, right? Stochasticity is there. Okay. In our simulations, we have that stochasticity. But for purposes of making an analytical calculation, and that stochasticity is small. I mean, you can see that in those curves. For purpose of making analytical calculation, we'll say that even though the birth and division volumes, they are going to be stochastic and governed by the Z dynamics, between birth and growth, the deterministic differential equations is what is going to guide them. And so, if you do that, then you see that the Z dynamics becomes very simple. We can now assume that R is RB e to the mu t, P is PB e to the mu t, V is VB e to the mu t, right? And then this whole thing becomes some constant C into e to the mu t, right? Where this constant is nothing but Kz into RB PB divided by VB. Is this point clear? But this dynamics, the Z dynamics becomes, the deterministic part of the Z dynamics becomes very, very simple. And let us take this chemical dynamics for the Z molecule and look at the stochastic version of this, because Z, the numbers are small. And while this is what would have been the deterministic dynamics of the Z molecule, this is the dynamics that needs to be treated stochastically. Okay. And so you can write down the master equation for the Z molecule using this h of t, and you can solve it exactly with the boundary conditions that at t is equal to 0, z is equal to some zb. Okay, in particular, zb can be 0, but you can solve it for arbitrary zb, and then it satisfies this master equation. And so this P of Zt, what is P of Zt? That at time t, there are exactly this many molecules of the that probability is what satisfies the master equation, and you can solve this. Okay, so I can discuss this first passage time problem. It's very simple, and you can actually solve this exactly. That's right. Yes. Yes. It is, but yes, that would be very difficult to solve. But this is, but because the stochasticity in the is ultimately going to be governed by numbers of those molecules, you can ignore that stochasticity. It's justified to ignore that stochasticity. Would be essentially the V would have a Z, let's say, and that would be stochastic, but that is going to contribute very small effects. Okay, governs the effective dynamics of the division sector, of the timekeeper molecule sector, because look, this function depends upon them, and as you can see that this constant then depends upon what is happening there in that sector. This mu depends upon what's happening in the sector. The mu is determined entirely by that sector, entirely by the PTR sector. So the Z dynamics is being affected by that sector, and the Z dynamics is in turn affecting the growth sector through division. Okay, so what the value of PB, RB, VB, etc. are going to be is being determined by how long it takes Z to actually grow to its threshold value, and that is what is going to determine the division, the division volume, and hence then the next generation's birth volume. And so it's cleverly done. It's cleverly done here. So that one can solve this problem. But these are two coupled dynamics, okay? One deterministic and one stochastic. Yeah. So that is what allows us to analytically solve it. But of course, you can treat both the dynamics stochastically. And so once you have the first passage time distribution, you see, the added volume is just this. The added volume is just this, right? And this is stochastic, so the added volume is stochastic. VB is stochastic, so but you can work out the first pass, the added volume distribution, and you can show analytically that it only depends. Okay, so this distribution is given by this, and look at. So the only parameter that appears in the added volume distribution is this parameter A, which is this, where C was given by this formula. Notice that C has two extensive numerator and one extensive quantity in the denominator. Okay. So as you scale the size of the system, it's an extensive quantity itself. It scales extensively. And what is appearing, this parameter that is appearing in the added volume distribution is C divided by mu B, by VB, and mu is again an extensive quantity which only depends upon the ratios of chemicals. So this extensive quantity and this extensive quantity, they cancel each other. Okay. And therefore, A is independent size of the system. Okay, it's independent of the work. And that is the origin of the adder property, that the distribution of added sizes is independent of the birth size. That's good. And of course, if you rescale the distribution, rescale the added size, and define a variable u, which is the added size divided by the average added size, then the distribution of this rescaled variable, the rescaled added size, is just given by this, where this n, I haven't written that down. I should have mentioned that. What is this n? I think I mentioned it earlier. Did I? Oh, I didn't mention it here. That is the most crucial parameter. This n is nothing but ZC minus ZB, which when ZB is equal to. I had mentioned it earlier. Yes, yes. Right. Okay. So the only parameter of the model that determines the added size distribution ultimately is ZC. Okay. So that's the. And so this is also a proof. I mean, by the way, this kind of behavior, this kind of equation, doesn't matter whether you did a PTR model or you did a PTR FVL or complicated model with n variables. As long as it is homogeneous degree 1, you would end up with these guys growing exponentially, and therefore you will end up with a Z equation which will look like this. Okay? It doesn't matter whether it is a PTR model. Okay? You could have any complicated nonlinear model as long as it is homogeneous degree 1. Okay? And the Z dynamics is also given by a homogeneous degree 1 function of the axis. You will always end up with this. And therefore, the Z part of it, the first passage time distribution, the added volume distribution, will always be the same and will only be governed by one parameter, which is ZC. It will be independent of all the parameters of the PTR sector. So this is not only, so this is a proof of universality of that dynamics. Okay. So through of all models. Okay. And so you can take this distribution and compare it to the experiments, and here it has been compared. So the dots, of course, are the experimental curves from this work, and the green line, which is a smooth line, is this formula. Okay. And the red curve, which is a jagged line, is our numerical simulations. So the numerical simulations, which include the stochasticity of PT and R also, in addition to Z, first of all, they agree with the green curve, which is based on an approximation in which the PTR dynamics is treated deterministically. Okay? So the red curve and the green curve agree with each other, and they agree with the experiments, so with this kind of value of Z. That's the thing. And you can, of course, explicitly, you can do simulations and have thousands of divisions and look at that. And for every generation, every cell, you can have one point in which you are measuring the volume at birth, the interdivision time, the volume at division, and delta. And you can plot those, make those scatter plots for each cell, just like it has been done in the experiments. And you see that the interdivision time is correlated negatively, the division volume is correlated positively, and the added volume is uncorrelated with the birth volume, as turned out in the experiments. Okay. So that is what I wanted to say about how one might understand the variability between cells. So notice that we are talking about cells which are identical to each other in the sense that their parameters are identical. It's the same Kp, K, Mt, Mr. The cell is exactly the same cell. It's being placed in the same environment. Kp does not change. Okay. And yet, individual cells have different sizes, different birth interdivision times, and so on. Okay. That's what is observed in the experiments. And a model like this reproduces the cell-to-cell individuality, and the mechanism is imprinting molecular level stochasticity, okay, in the population, in the chemical dynamics, which is very just natural. Okay, that ultimately is coming from just the stochasticity of collisions. Who collides with whom? And whether that collision leads to a reaction when they collide, this is all governed by classical, statistical mechanics and quantum statistical mechanics. Right? These are that stochasticity alone in such a scheme can be responsible for the cell-to-cell individuality. And the amazing thing, to my mind, what I find very striking is that there is this typical CV. Okay, of 10 to 30% that you see in all these distributions that you experimentally get right from the beginning of the time that these measurements have been made. And that has just a simple explanation in terms of the small size between 10 and 100, okay, of this threshold to which this molecular thing has to go. So that is where the stochasticity is coming. That is where this 1 over root n factor is coming in, which is governing the width of the distributions. And that is imprinting itself onto a macroscopic fluctuations of the cell. Okay, so that's, in this, I have 5 minutes. Yeah, okay. So I will just mention a couple of things now briefly. Well, there is one experiment that has been done, more than a decade and a half back. And there does not seem to be any explanation of this experiment. And the experiment is the following: that you take E. coli cells, again, in the same medium, in a fixed medium, fixed strain, and you somehow tag a very large number of proteins inside the cell simultaneously and measure their populations. Okay? So you just measure the fluorescence of a large number of proteins inside the cell. Okay? And so, you find proteins, some proteins are present in large numbers, the mean number of proteins that they measured, the fluorescence was, let's say 10 to the 3,000. And some proteins are present in very small numbers, okay, of order 1, okay. The average might even be less than 1 across cells. Sometimes you see the molecule, sometimes you don't see the molecule, okay, etc. So you have, several orders of magnitude in terms of the average population of the protein. And depends on which protein you're talking about. Some proteins are present in small numbers, some proteins are present in large numbers. Each dot in this curve corresponds to a protein measurement. And now, since you are measuring it across many cells, you can also look at the variation of the protein number for any particular protein. You can also measure the variation across cells. Okay? So the protein copy number is not the same in all the cells that you measure it, and there is a distribution for any particular protein. This is for this particular protein. You have a distribution of protein numbers across different sites. Now, what they've done is that they've plotted the variation versus the mean for every protein. And you get a scatter plot like this, which seems to be on this log plot, a sloping line with negative slope, with actually a slope -1, okay? For small numbers, for proteins whose mean numbers are small, and then it flattens out. Beyond a certain protein number, it doesn't matter. They all have the same variation. Okay? And this was considered to be an extrinsic noise. You see, so this is the intrinsic noise. Intrinsic noise is if you have small numbers, then just 1/√n. Right? So the fluctuations are 1/√n, and therefore the variance, that is the CV will be 1/√n. The standard deviation squared divided by the mean, which is the variance divided by the mean, will be 1/n, right? 1/n, and where n, the mean is n, so it'll go as with a slope -1. Okay. But then when the numbers become sufficiently large, it flattens out. And so this was thought to be some extrinsic source of variation. But in the model that I just presented to you, you can add some protein, Q dot, with this kind of a garden variety dynamics, and make it stochastic. And different values of KQ will give you different means. So you look at proteins with different values of KQ. And when you do that, what you find is that the variation looks exactly like this curve. And you understand that when the protein number is very small, then the intrinsic fluctuations dominate. But this flattening out is because, the fluctuations, when this number of proteins is larger than ZC, well, then the fluctuations of ZC are what are going to dominate. Right? So that is, in every cell you will get fluctuations of this number just because, the value of ZC in the previous generation was different. Okay? So the extrinsic noise is explained in such a model as a fluctuation of the first passage time of the timekeeper molecule. Okay? That's another part that gets explained. Yes? Yeah, sure.
So the fluctuations in all the other proteins which are high in number are a function of your timekeeper molecule Z.
Right. That's right. So they're dependent on ZC. And ZC is fixed.
ZC is fixed but a small number, and the fluctuations are arising because of the smallness of ZC.
Yes.
So are you claiming that in this observation by Taniguchi,
yes,
the variation for proteins which have high mean protein number is occurring because of that small because of the intrinsic noise of the small protein number molecules?
That is also there. That fluctuation is always going to be there. But because their numbers are larger than ZC, that number is going to be dominated by the fluctuations that are going to arise because because of this ZC issue, that in every cell it's the ZC of the previous generation that's going to determine what how much protein was produced in the previous generation.
Right.
So this explains why there is a crossover. If the protein population is less than ZC, its intrinsic populations will dominate. If the protein population is greater than ZC, then the ZC, its fluctuations will be dominated by this effect that I described earlier. That's what I'm saying. Okay, so I will now. Oh, I just want to mention that recently we've just looked at division more carefully. Actually, it was done much earlier, but wrote it up recently. And we find that division can have some very non-trivial effects, okay? Including, even for class 1 systems, you can get non-exponential growth and you can get unbalanced growth. Okay? So I just want to mention this. This has not been noticed earlier. So there is a geometric understanding of what is going on in terms of simple models, but I don't want to get into that, and I will just flash this summary slide at the end. Okay? So I just leave it at that and let you ask the question.
So I didn't get it right, probably the extrinsic fluctuations. So you are measuring the CV of the protein molecules, right?
Yes.
And are you suggesting the fluctuation in timing somehow feeds back to this CV of P?
That's right. Exactly. That, how large, in the previous generation, you have a certain cell. Of course, this cell is going to divide at a certain time, which is going to be determined by the first passage time that you will get in this generation. That is, whatever the Z, however long it takes Z to reach ZC in this generation. But what is the PD that you started with? At birth, what was the population of the P molecule, the R molecule, the T molecule, etc., or any molecule, Q molecule actually, that you started with? What is the population that you started with? That depended upon the birth volume, the birth value PB, okay? This PB was half, so the birth value in the GH generation was half of PD of the G-1th generation. And the fluctuations of this guy were determined, how large this was determined by, so PD in the GH generation was PD in the GH generation multiplied by this factor, tau in the GH generation. And tau in the GH generation was controlled by the Z dynamics of that generation. Okay? So I should be talking here about G-1. This is all G-1. This is G-1. So, I'm looking at the fluctuations in PB in the GH generation. But that depends on what you started with so PB in the GH generation is half of PD in the G-1th generation. And this division depended upon PB in the G-1th generation multiplied by this factor, which is stochastic. And its stochasticity is controlled by the first passage time of ZC in that generation. Okay? And it's the CV of that tau, and hence the CV of this guy, that is this guy, is controlled by that. Okay? So that is why when your cell was born in the first place, there was a fluctuation in PB. That is how it is. It is governed by this by the timekeeping.
But generally, extrinsic noise are considered to be from global sources, right?
Exactly. So this is an intrinsic source of extrinsic noise. Okay, if you like. Okay. So this is an explanation of extrinsic noise based on an intrinsic stochastic fluctuations of a molecule. You don't have to break anything outside.
Right. And one thing, like, and it's a global thing, yes, I think. So we learned that the growth rate is endogenously selected. It's kind of maximized by the cell depending upon the global situation. So Bialek has an interesting proposition that cell generally tries to maximize or optimize information content depending upon its interaction with environment. And there are like some forbidden region that some of those ways are not accessible to cells. So what's your like general comments on that?
So I don't have very much to say about it, you know. It's probably true. Okay. I don't, what I have said, I think, I don't think sort of either contradicts or supports any of that. Okay. So, so that is, you know, so the optimization that I have been talking about is something that is, of course, evolutionarily generated. Of course, I'm sure in all the examples that Bill Bialek has in mind, the fact that the cell is optimizing its evolutionary dynamics, optimizing information extraction from the environment, let's say, is also evolutionary generated. I mean, the design is evolutionary. And so to that extent, you know, this is also, you know, in the same sort of vein that the optimization is a consequence of evolutionary mechanism. Okay? But here, there's no information. I mean, you know, there's no information theoretic sort of component of whatever I have said here. Okay. Maybe there is a way of thinking about it in terms of information. I haven't really thought about it.
Cells often sense their environment, like whether it's a nutrient-rich medium, say it's a LD medium or a starvation medium.
Yes.
So according to which it will, you know, tries to tune its new.
Yes. Yes. So what I can say is that I had a model in which the growth rate was not happening due to optimization, but was actually a dynamical variable, which was affected. There was a mechanistic model that affected the growth rate, right? And so that mechanism was that. So there was a sensing, okay? So there was a certain regulation there, and that regulation obviously involved sensing. You know, so that of course is happening, but there is no stochastic. I mean, that was a completely deterministic model in which you could see that optimization coming up. So there was no, stochastic fluctuations there. There was a mechanism. There was a explicit dynamical mechanism which was giving rise to that sensing. That sensing mechanism, if you like, was put in by hand in the model. So that's what I would say. Well, maybe there's something that you're trying to get at. Maybe we can talk about it later. Okay. So
yeah, like, so the idea was that depending upon the medium that you expose those cells in, the bacterial cells are, so they can actually sense the environment and thereby that tune its growth rate to maximum level that it can.
yes.
So that was the thing. I think
that is true, and that is happening here in this model. That is certainly true. I mean, there's no doubt about it.
This part is clear, but the part about Bill Bialek was not clear.
Yeah, like the information exchange between the environment and the cellular components, right? The concentrations of these molecules and the extrinsic molecules that it senses, for example, in the growth media. So that information, say the mutual information, and actually Bialek has a paper where he has like connected into the growth rate of E. coli cells in different conditions and showed that it always tries to maximize the information content. No, it was kind of, unpublished work. It remained on archive from 2007, I guess. Yeah, it's a short paper, yeah.
It is very clear that the body will optimize. I am not sure that you can capture the rich from tissue or not. It captures the richness of the medium. So this is the model that captures the richness, which senses the richness of the medium. Maybe in interest of time, we can talk about it offline, and I would like to also see this paper. But clearly there is a sensing involved here. But this sensing in this model is completely deterministic. Okay? I can of course make this model stochastic. Okay, like I made the PTR model stochastic. We can make this model stochastic, and maybe there are some questions there that one might that might be interesting to look at. So maybe we can talk about it.